Hello viewers, in the last lecture we discussed about necrosis and its various patterns. In this video we will be studying about apoptosis. This is video 1 of apoptosis, in which we will be discussing definition, causes and morphology of apoptosis. In video 2 we will be studying about the mechanism of apoptosis. So what is apoptosis? Apoptosis is derived from a Greek word that means falling off. Like falling off of leaves during autumn. So what is the definition of apoptosis? Apoptosis is a type of cell death that is induced by a tightly regulated suicide program. What does it mean? It means that it is regulated at molecular level. There are certain genes that are regulating it, in which there is activation of intrinsic enzymes. What are these enzymes? These are caspases, in which there is activation of intrinsic enzymes that cause breakdown or degradation of genomic DNA, nuclear proteins and cytoplasmic proteins. So in apoptosis, what happens is the cell is broken down into apoptotic bodies. Apoptotic bodies. What are these bodies? These are nothing but cellular fragments. These are fragments of cells. These are bound by plasma membrane and they contain cellular organelles. They contain cellular organelles which are tightly packed in these apoptotic bodies. They contain cytoplasm and they may or may not contain the nuclear fragments. So what happens in apoptosis? The plasma membrane is not disrupted. It is intact, but there is some structural change in the plasma membrane, like the phosphatidylserine that is present in the inner leaflet of plasma membrane. It gets flipped to outer side of the plasma membrane. So in healthy cells, the phosphatidylserine is normally present towards the inner side of the plasma membrane, and in cells that are dying by apoptosis, it gets flipped towards the outer side. So these types of cells or these types of apoptotic fragments, they are easily recognized. They are easily recognized or identified by macrophages. So they are recognized by macrophages and they come to eat up or clear up these apoptotic bodies. So they are phagocytosed by macrophages. So what does it mean? It means that there is no spillage of the content from the apoptotic bodies, so there will be no inflammation. So there will be no inflammation. Whereas in necrosis, inflammation was an important feature. So inflammation was also causing tissue damage. So there will be here in apoptosis no inflammation, no tissue damage. No tissue damage is seen. And necrosis was seen in pathologic conditions. The cause of necrosis was pathologic, whereas apoptosis can be seen in physiological conditions also. So it is seen in both physiological situations as well as in some pathologic conditions. It is a mode of cell death. So now we will be discussing about what are these physiologic conditions and pathologic states in which apoptosis is seen. So what are the causes of apoptosis? Apoptosis is seen both in physiologic situations and in pathologic conditions. So what are the examples of apoptosis that is occurring in physiologic situations? So in a human body, the cell turnover is 1 million cells per second. So how this is happening? This is happening because of apoptosis and by the action of macrophages on the apoptotic bodies. So first is the removal of supernumerary cells, the cells which are present in excess of the required number during development. So in this there is involution of primordial structures, and during remodeling in maturing tissues. So when cell death is occurring in the context of development and embryogenesis, we use the term programmed cell death. Programmed cell death. And when we are talking about this pattern of cell death, we use the term apoptosis. So programmed cell death is used in the context of cell death or removal of cells that is occurring during development, during embryogenesis. Second is involution of cells, involution of hormone dependent cells in the hormone dependent tissues, on hormone withdrawal. So what is happening here? This survival signal, the hormone, we are withdrawing that, so cell will die. So example is the regression of lactating breast on weaning, the breakdown of endometrial lining during menstruation. Another example is cell turnover in proliferating cell populations. So the cells that are actively dividing, for example, the crypts of intestinal epithelium, the crypts in the intestinal epithelium. So these cells, they are actively dividing. So to maintain a constant number of cells or homeostasis, these cells undergo cell death. Removal of potentially harmful self-reactive lymphocytes. So what happens? So these self-reactive lymphocytes, they can produce immune reaction against one's own cell. So removal of potentially harmful self-reactive lymphocytes occurs by apoptosis. If the mechanism of apoptosis is defective at this level, what will happen? It can lead to autoimmune diseases. Fifth is removal of host cells that have served their useful purpose. For example, neutrophils, lymphocytes, what happens to them at the end of immune response? Neutrophils, lymphocytes, they at the end of immune response, they die by apoptosis. So what is happening at the molecular level? So these cells, they are deprived of survival signals, which are in the form of growth factors, hormones, and interactions with the extracellular matrix. Also, there can be increase in pro-apoptotic signals from the neighboring cells or the environment. Next, we will study about the pathologic conditions in which apoptosis is seen. So next is the pathologic causes or the pathologic conditions in which apoptosis is seen. So it is seen in those cells that have been damaged beyond repair. There is no inflammation, so the cell undergo apoptosis. There is no tissue damage. So what are the examples? When there is DNA damage. So DNA damage can occur due to radiations into cytotoxic drugs. They can directly damage the DNA, or it can be damaged due to production of free radicals. So what happens when DNA gets damaged? The p53 dependent pathways, they come into play. p53 dependent pathway, they try to repair the DNA, and when this repair mechanism fails, then p53 triggers cell death by apoptosis, by the mitochondrial pathway. So it is better for the cell to die which has undergone DNA damage. Why? Because otherwise it could have led to malignant transformation. So apoptosis acts as a preventive mechanism. So the cell that has incurred DNA damage, it dies by apoptosis. If this p53 dependent pathway, it is intact, either it tries to repair it. If it fails, the repair mechanism fails, then it triggers apoptosis, which is beneficial for the organism. Next is accumulation of misfolded proteins, which leads to ER stress, endoplasmic reticulum stress. So this also triggers apoptosis. Then infections, particularly viral infections, they induce apoptosis either by themselves or due to host immune response. So due to virus or the viral infection, example is HIV, adenovirus, and due to host response, the apoptosis is seen in viral hepatitis. The cytotoxic T lymphocytes, cytotoxic T lymphocyte, they play important role in inducing apoptosis of the viral infected cells. So in a way they remove the reservoirs of infection. They remove the reservoirs of infection. They remove the reservoir of infection. And they also play important role in killing tumor cells and in the removal of or the rejection of transplants. Next is the pathologic atrophy of parenchymal tissues or organs after duct obstruction. This is seen in pancreas, the salivary glands. So when there is duct obstruction, the tissue undergoes cell death or it regresses in size due to apoptosis. So next we will study the morphology of apoptosis. What are the morphologic changes in apoptosis? These are best seen with the help of electron microscope. These are cell shrinkage, cell size is decreased. This is in contrast to that seen in necrosis where there is cellular swelling. So the cytoplasm is dense, eosinophilic, and the organelles are tightly packed in the cell. So how it appears? This is the cell. The cell is shrunken in size and it appears deeply eosinophilic with nuclear fragments. Second is chromatin condensation. So this is an important feature, chromatin condensation. So there is chromatin condensation and the chromatin gets aggregated. Chromatin aggregates beneath the nuclear membrane. In various shapes and sizes. And later on the nucleus gets fragmented in 2 or more fragments. Nucleus gets fragmented. So here these are the fragmented. This is the fragmented nucleus and this is the dense eosinophilic cytoplasm, So suppose this is the nuclear membrane. So the chromatin, it aggregates into various shapes beneath the nuclear membrane, and later on the nucleus gets fragmented into 2 or more fragments. Third is formation of cytoplasmic blebs and apoptotic bodies. Plasmic blebs and apoptotic bodies. So first there is formation of cytoplasmic blebs, and then this leads to formation of apoptotic bodies, and the cell gets fragmented. So these apoptotic bodies, they are lined by plasma membrane. They have cytoplasm, they have organelles, and they may or may not contain the nuclear fragments. Fourth is phagocytosis of apoptotic cell or apoptotic bodies by macrophages. So the apoptotic bodies, in these the phosphatidylserine is expressed on the outer aspect of the membrane. So these are easily identified by the macrophages and they are rapidly cleared by them. So the lysosomal enzymes in the macrophages, they degrade these bodies, degrade the apoptotic bodies. So that is why they are not easily seen on light microscopy. The apoptotic bodies are being formed rapidly, and they are being rapidly cleared up by the macrophages. So apoptotic, the morphology is best appreciated with the help of electron microscope. So in the next lecture or the video 2 of apoptosis, we will be studying about the mechanism of apoptosis.